Thank you, Colin. Good morning, everyone. I'm Choi from UCLA. Today I'm going to talk about a new project, MetProt, which is a cloud-based platform that analyzes, annotates, and integrates two omics, metabolomics and proteomics. This is relatively recently launched project, so in today's presentation, I will mainly go over the specific aims of the project as well as our approaches. And at the end of this presentation, I will show a quick demo. In the past few years, I have spent a lot of time to learn the proteomics. However, every person I talked to gave me a different definition of what is a proteomics. And also, since 2 years ago, I'm actively involving the metabolomics study, and every books and articles that I have read gave me a slightly different answers that what is the metabolomics. So my decision here is I will take the common sense. So I consulted with the Wikipedia. So here's the definition from Wikipedia. So proteomics is the large-scale study of proteins, particularly their structures and functions. And proteome is the entire set of proteins produced or modified by an organism or system. Metabolomics is the scientific study of the chemical processes involving metabolites. And metabolome refer to the complete set of small molecules metabolites, such as metabolite intermediates, hormones, and other signaling molecules, and secondary metabolites to be found within the biological sample, such as a single organism. And both proteins and metabolites are like a very essential building blocks of cellular processes that collaboratively formulate biological pathways. So in the Aztec Discovery Index, we were able to find 1,020 proteomics tools in databases, while we only found 32 metabolomics applications in databases. And to our best knowledge, the availability of application that comprehensively analyze, integrate both omics is currently very limited. And this limitation hindered our capability to identify biomolecules relevant to the really relevant to the certain disease. So the MetProt project is a simple and straightforward. So first, understand each concept of omics to be able to analyze complex data sets. And second, annotate the data sets, not just give the simple description of data, but provide data sets functional meaning and their relationship with other information. And finally, integrate the proteomics datasets with the metabolomics dataset through shared interfaces between protein and metabolite. So analysis, annotation, and integration are the key concept of this project, and those will be achieved in the following three specific aims. So aim one is to develop a simplified computational pipeline that mainly identify driver molecules from large-scale time series omics datasets. And to accomplish this, we are utilizing two datasets. One is a dataset from the mouse model of heart failure, which will be introduced in later slides. And the second is the datasets from heart failure patient, which will not be shown in this presentation. Aim two is to develop a bioinformatics strategy to integrate proteome datasets with metabolome datasets. In this aim, we first identify where protein meets metabolites, and then establish the bioinformatics strategy. to be able to test this, we will utilize both proteome and metabolome datasets obtained from the 6 genetic mouse strains. And lastly, aim 3 is to establish a cloud-based platform to enable and disseminate the computational pipeline and bioinformatics strategy developed in aim 1 and aim 2. So in aim 1, I will briefly talk about the temporal data sets we have acquired in an analysis pipeline simplified analysis pipeline. Let me quickly introduce time series data sets acquired from the mouse model to human cardiac disease. So this panel illustrates the schematic overview of the experimental design. And when the mouse model is under the cardiac stress, it develops hypertrophy. So to do so, mice continuously received isopretonolol through the pump through up to 14 days. And plasma and heart tissue sample were collected 1 day before the treatment and several days after the treatment. And along with these sample collection, we have measured the phenotype, the heart rate body rate ratio, which indicates condition of heart. And we replicated the experiments in 6 common genetic mouse strains to make sure that the computational pipeline that we aim to develop, the computational pipeline that we are developing is more likely applicable to the heterogeneous data sets. And currently, the plasma metabolome and cardiac proteome data sets are available, and we will later collect the mouse cardiac metabolome data sets. So this diagram is a schematic overview of computational workflow. It takes time series. I don't know whether you can see my cursor here. So it takes time series omics data set with the phenotypic information as input, and the pipeline mainly consists of 3 layers, which is a cleaning, and analyzing and verifying, and the output is the identified driver molecules. So we first implement a data preprocessing module that basically clean and normalize as well as organize the input data sets so it can be easily adapted in the following analysis. And the cleaned data is then analyzed by 3 main modules in here. The differential expression analysis is basically aimed to detect significantly altered metabolite over time. And the linear mix model is it identifies metabolite that are strongly associated with phenotypic characteristics. And the pattern recognition is aimed to identify the temporal pattern of the molecules, and it clusters the molecules based on their dynamics. and later, the metabolite that are detected and characterized by those 3 modules will be subjected into the principal component analysis and hierarchical clustering to be visually verified. So if the results indicate that the commonly detected molecules have sufficient power to stratify different conditions, and those molecules will be considered as driver molecules. So I will give one quick example of the pattern recognition. So by utilizing our pattern recognition module, we were able to identify 6 distinct dynamic patterns of metabolite profiling from mouse datasets. So we have identified those 6 different distinct patterns. Pattern 1 is a stable. Pattern 2 is random, which means it fluctuates a lot. And pattern 3 is increased. Pattern 4 is a monophase increase, so basically increase after the treatment and decreased in going back to the normal phase. And pattern 5 is decreased, and pattern 6 is a monophasic decrease. So here's a preliminary.
Hi, this is Jose. Can I ask you a question on the slide that you just showed? Are the time there, the time points? Maybe sorry, I missed the first part of your presentation. What is the time there in these patterns? Are you talking about minutes, hours?
It's a days.
These are days. It goes from day 1 to.
Yeah. So basically, in our mouse experiments, it's basically mice received the isoproterenol for up to 14 days, and we collect a sample from 1 day before the treatment and 1, 3, 5, 7, 14 days after the treatments. So it's basically, yeah, I'm sorry, I didn't label here, but like it's a 0 days, 1, 3, 5, 7, 14, and 14 days.
Okay, thanks so much.
So here's the preliminary results of the pattern recognition in 3 different mouse strains. And as you can see, this lipid metabolites show the similar trends over the 3 different mouse strains, and it's basically recognized as a pattern 5, which is a decrease. While the alanine showed a similar trend in FEB and C57, which is a pattern 6, but it shows a very different patterns in BALB-CJ mouse strains. So it basically increased right after the treatment. And interestingly, the FEB and C57 are known to be resistant to the ISO treatment, while the BALB-CJ is sensitive. And the phosphatidylethanol ethanol amine show the distinctive pattern across the 3 different strains. So it's more stable in FEB. It's a monophasic increase in C57 and continuous increase in BALB-CJ. Okay, so let me move on to the AIM-2. So the topic of AIM-2 is the annotation and integration of metabolomics and proteomics datasets. So the rapid development of analytical technology has drastically increased, and amount of data generated by both proteomics and metabolomics experiments are very high. However, many large data sets of either proteins or metabolites are often fragmented and rarely integrated. So in the AIM-2, we will develop the strategy to integrate metabolomics datasets with the proteomic datasets to address the issue. So first, we study where metabolome meets proteome, and we identify 5 important relationships between proteins and metabolites. First, they're shared biological and cellular pathways. Second, chemical and biochemical reactions. Third, post-translational modification for metabolite as ligand for protein-based receptor, and 5 metabolite-based transcriptional regulation. And the databases in the parentheses are the database we will utilize to find the interface between protein and metabolites. So I would like to use a particular biological process that consists of multiple biochemical reaction as an example. And this scenario, we have unacetylated proteins with the free acetylated proteins as well as a major. Just give me a second. Yeah. So do you see my cursor? Can you see my cursor?
Yeah, we can see your cursor.
Okay, so here's the unacetylated proteins and with a free amino group here, and with a histone acetyltransferase, the acetyl-CoA here is transferring to the histone protein, and the acetylation is considered. And this process, this acetylation is considered as the post-translational modification. And the second, the acetylated proteins can be deacetylated by the histone deacetylase. Third one is the byproduct of the acetylation, which is coenzyme A, can be recycled to generate acetyl-CoA through the glycolysis pathway. And the acetyl-CoA can be consumed during the TCA cycle pathway to produce coenzyme A. And fourth, there's the broad spectrum of drugs which can be inhibit the function of histone deacetylase. And finally, the acetylation will active histone, which will contribute the transcriptional regulation, which is the indirect regulation. So accordingly, we identify the following relationship among proteins and metabolites. So first, we found the biological pathways and the chemical and enzymatic reactions and post-translational modification and ligand and receptor as a drug target, and basically indirect transcriptional regulation. And so we believe those are the interfaces where the proteins meet metabolites, and we will retrieve this relevant information from multiple databases and knowledge bases, which will be actually specified in the later slide. Okay, I will move on to the aim 3. So aim 3 is about building a cloud-based platform. So this diagram shows a MetProt's systematic architecture, and user will able to access MetProt through the either RESTful API or website. Once users submit a query through those access points, MetProt core will analyze, annotate, and integrate the input data sets, and information archived in the MetProt database will be accessed through the DAO. And we will implement the MetProt database updater in Java, and this component will integrate various types of biomolecular interactions informations from Reactome, CAG pathway, UniProt, HMDB, DBPTM, Phosphosite Plus, CAG ligand, and drug bank. and it will store the comprehensive interaction information among proteins and metabolites in MetProt database through the DAO. In addition to the MetProt, in addition to that, the MetProt will provide biological and chemical functional annotations for protein as well as a metabolite, and by utilizing APIs provided by GeneWiki, BioGPS, Metabolite, HMDB, KEBI, and CopaKB. And MetProt will be hosted in Amazon Web Service EC2 server. Here's the diagram in the schematic overview of the integrated workflow. User will be submit in two major categories here, which is one is a list of proteins or metabolite identifiers, and the other form is a large-scale molecular data sets with quantitative value as well as the phenotypic information. So for the latter category, MetProt will review and analyze the data to identify driver molecules that significantly altered and highly associated with the phenotypic characteristics, which is achieved by AIM-1. So in AIM-2, the molecules of interest actually detected through the AIM-1 will be explored using information fetched from pathway, chemical reaction, PTM, ligand, drug, transcriptional regulation databases. And the extracted information are now integrated to construct network graph among proteins and metabolites, and with additional annotation of molecules from CopaKB, GeneWiki, and Metabolite and KEBI. So collectively, we will engineer a cloud-based platform to enable this analyses on the cloud and to generate integrated report, including table view of protein metabolite interaction, functional annotation, and graphical views as output. So in summary, we are establishing a cloud-based platform called MetProt for triaging and analyzing omics data sets and finding existing as well as novel connections between proteins and metabolites, and annotating molecular functions and providing biomedical insights. So I will give you a very quick demo about the AIM-1. And in AIM-1, the followings are the technology used. So we have Java on the back end with the Spring and Spring Boot for the server, and our Java is utilized to run our sublets on the back end. So all the analysis is basically done by R. And the database, we were initially using the MongoDB, but we actually now we are using DynamoDB since our server is hosted in Amazon, and DynamoDB is an Amazon product, so it's more reliable and easier to maintain. Third, the front end, we use HTML, CSS, and JavaScript Bootstrap, and we use D3.js for the graphics. It's going to be very quick since we recently launched the project, but at least I can show the quick demo so you can see where we are. So this is the look of the front, the main gate page. So you have the button for the analysis, and you have a 3-panel that shows the recent updates, the user, the feature user statistics, as well the site traffic. And if you to start analysis, you can just click the start analysis, and all the functionalities that I mentioned in the presentation are listed on the left side, and it will take the list, the input file, which contains the quantitative value as well as the phenotypic characteristics. And it will do quick integrity check, so it will tell you the number of inputs as well as the number of missing values. And you'll be able to see your input data on the right side. So you'll be accessible. It's accessible anytime. So once you upload the data, you'll be able to do the pre-process. You can handle the missing data, as well as you can select the method for normalization, transformation, and data scaling, and you can process. Then it will automatically, spontaneously create the pre-processed file. And if you go to analysis, so far we have 2 components implemented, so differential expression analysis and pattern recognition. So in the differential expression analysis, it performed the t-test and utilized a fold change to create the volcano plot, and all the upregulated and downregulated molecules or either protein or metabolites will be listed in here. And by clicking each molecule, it will highlight the molecule that by the click. Also, you'll be able to zoom in, zoom out, as well as you can change threshold as well, and it will spontaneously change the list of the upregulated and downregulated molecules as well. And you'll be able to download high quality SVG file, so you can use this graph. You can directly use this graph for your publications. In pattern recognition, it's basically so you need to specify the number of desired cluster and the minimum number of metabolite per cluster. So it identifies 3 clusters, and by clicking each metabolite, you'll be able to see how the pattern of the molecules over time. And also, so in the data sets, the data set is for 3 mouse strains, and you'll be able to see the individual, the pattern of the metabolites for individual mouse strains. So let's say the alanine. So alanine shows very similar patterns in first strains and second strains, while it shows very different patterns in third strains. So we will keep working on the MetProt, and I hope to give like a solid demonstration in the next technical conference call. And that's pretty much it. And thank you for listening, and I'm happy to take any question. Thank you.
Thank you, Howard. Do we have any questions?
I have one simple question to start with. I tried to connect to metprot.org, and the printer it's to me at Annotate is blocked. Is it active to the public now, or?
No, it's alpha version, so it's not active now.
This is Andrew Su here. Nice presentation. I have a question on this, actually the experimental design you're showing here, the 3-strain analysis. Can you talk a little bit about the motivation of doing that? And I assume to some extent you're trying to correlate proteomic and metabolomic variation to phenotypic variation. But of course, like having only 3 strains is a challenge as well. So anyway, if you could just talk about sort of how you're thinking about this experimental design.
So basically, we thought there's a limitation of the before and after study. So although like some molecules has a drastic change like before the treatment and after the treatment, but if you look at the very long term, actually sometimes, although like in before and after study it shows a significance, but in actual, if you look at the actual patterns over time, over several time points, we actually found those metabolites are not actually significantly different. So we thought the time series data set can give more the deeper insights about what actually the biological processes. But there's a method, but most of people actually are doing is with the time series data is just a simple clustering based on the pattern. So actually, the computational pipeline in MetProt will really more focus on the time series data sets analysis. Did I answer your question?
I guess I was specifically interested in how you were going to interpret the strain versus strain similarities or differences. Your comparison between C57 and BALB-C and FVB.
Yeah. So first, the way to do it is as I presented in here. So we set, so basically it look up, so we have to set the threshold based on the distribution of the data, and then classify, and then like we have to come up with different patterns. Different patterns of the metabolites and then and go over the metabolites of the each strains and actually assigned the metabolite with the number, pattern number. So you'll be able to see the metabolite from FEB is actually pattern 2, and this metabolite from the C57 is actually pattern 3. So you'll be able to say they have like the metabolite follow a different pattern. It's yeah, that's the one way what we're going to that's the thing what we are going to implement in the analysis.
Got it. Thank you.
So this is Henning. Hi, Howard. Very nice
Hi.
presentation.
Thank you.
Can you comment a bit on on timelines and who is we? Looking at AIM3 alone, I think I have seen fairly similar slides as you go a bit further as what you're showing on these slides as multi-PI, multi-year EU projects for data integration. So clearly you have a limited scope. You don't need to fully integrate. You only need to take what you need for your specific task. But nevertheless, is it multiple people, and what's the timeline?
So you're talking about the people that involved in this project as well as the timeline of the completion?
Yes, I just wonder, it's a lot of work, so I wonder what are the resources available for this, because certainly just integrating data from all these resources which you list on the slide is a very significant task. Or are you using a resource which has that already pre-integrated or so?
Yeah, so I'm fully supported by Dr. Ping, and so recently we hired the programmers for the front end, back end, as well as to do the data science as well. So the timeline for AIM1 is I believe I can give the full demonstration of AIM1 by the next conference call, which is probably 2 months later. And for the AIM2, so we will focus on mainly focus on the pathways and reaction database first. So the timeline, I had a slide for the timelines. The timeline for the full implementation of the pathway database and the retrieval information and organize the information from the pathway databases and reaction databases would be, if I remember correctly, it's August.
Okay, that's an aggressive timeline, but it's good to hear that it's a team behind that. So that's
yeah. We do have like a full team behind it.
Okay, good.
Yep.
Any other questions?
I have this is a more general question. I'm guessing at least most of us or some of us would expect that the data come out from metabolomics and proteomics, are not complete, either of them, and that you might have some complementarity, however, that would enhance the overall metabolism pictures that you can get out of it. So is there something that you're trying to do indeed to get the synergy out of the data?
So are you talking about, probably you're talking about the validation using the data sets from the proteomics and metabolomics, right? So we are trying to collect metabolites, metabolomics and metabolome and proteome from the same model, and the synergy that we can have is, just give me 1 second. So we could use those data sets for the validation of the strategy and the computational tool that we are developing. Since our ultimate goal is to find out the interactions and actual biological processes happening in the model. So we can use like the proteomics and metabolomics data sets acquired from same sample to validate our strategy.
Yeah, I mean, what I was trying to say that I think somebody managed to do in few cases, managed to actually use the model in complementary way, the proteomics data and metabolomics. That is, for example, in some cases, you might be able to assess the concentration of enzymes. In some other cases, that is not feasible, does not appear from the proteomics data. However, the concentration of metabolites is more readily available, and therefore you can make inference to try to have a more complete pictures of where the network, the metabolic network is.
I see. Yeah, we will think about it, and it's a great suggestion. Yeah, but we don't actually have like a solid plan. I mean, in terms of the very deep, finding very deep interaction between those two, but at least by this project, you will see where the molecule, I mean, let's say where my interesting metabolites interacts with the proteins. Is that, sorry, does that make sense? Yeah, but like we'll also consider what you have suggested.
I think I was just trying to see you were thinking that arena too or not.
Any other questions?